Okay, welcome to the last video in the series. This one is on the electron transport chain, a really important process that generates by far the majority of ATP in our bodies. The electron transport chain takes place on the inner membrane of the mitochondria, in structures called the cristae, which you remember are large folds of that inner membrane. So I'm going to show you the inside of one of those folds and some of the proteins that are involved in it. But first I want to show you that there is of course an outer membrane as well to mitochondria, which I'm going to put along the top like this. So there's my phospholipid bilayer, and that's the outer membrane. The inner membrane has got lots of large folds. Just to simplify this, I'm going to show you the dip of one fold and the proteins that are present in that. So here is the inner membrane coming in from one of these folds. Like so, there's my phospholipid bilayer, and in that membrane, and I'm going to exaggerate these just to make it really clear, I have a series of protein complexes embedded. So there's my first one, second. Now there may be a slightly different number to the number I'm drawing, but that doesn't particularly matter as long as you understand the principles behind it. So these protein complexes have various sides and they have different functions, which I'll explain in a moment. I recommend that you draw along with me as I draw. I think that'll help you a lot to understand what is happening in this process. Along here I have another protein complex. This one has an interesting shape, something like this. I'll just draw it out like so. On the other side, like this. Again, I'm going to do, coming down the other side, I'm going to do the other fold in of the cristae. It is important to remember that these protein complexes would happen many, many times all around the inner membrane of the mitochondria. So if you watched the last video in this series, you'd know that this is the matrix of the mitochondria. These are the cristae, and this is the intermembrane space. So let's label those. We have matrix, and then this is the intermembrane space. Remember, inter means between two different things. And then of course we have the outer membrane and the cytoplasm of the cell, also known as the cytosol. Outside. So we're looking just at one very small section of a mitochondria. Now, the Kreb cycle took place in the matrix, and the Kreb cycle produced a number of different molecules that we're going to need in our explanation of this. I'm going to talk about one of them first, the one that was mentioned several times. 4 molecules were produced per pyruvate in the matrix. That was NADH. Remember, we're talking about respiration here. This is the final step in respiration. There's no P. P in between the D and the H is only when we're talking about photosynthesis. So the NADH gives up its high energy electrons and is converted into hydrogen ions and NAD+. Now this will go off, it's come in from, let's just draw this on here and back. This has come in from the Kreb cycle, so I can just write that in here. Like so. Now NADH carries 2 electrons. So let's write this in here, 2E-. Like so. So it's given up its electrons and passed them onto the electron transport chain. And the electron transport chain is really referring to this chain of proteins in the membrane. And they literally pass the electrons from one to the other all the way down their length. There'll be electrons at every step here, so they'll all be moving together down the chain. Okay, so maybe it is a little bit like a train. As they move down the electron transport chain between these proteins, they lose energy, and that energy is used to pump hydrogen ions across the membrane into the intermembrane space, like this. So any hydrogen ions that are present here are being pumped across. This greatly increases the concentration of hydrogen ions, or protons as they're also known, in the intermembrane space. So I'm just drawing a large number of hydrogen ions on this side. This is a form of active transport, and it's a very similar process to the electron transport chain that you may remember from the video on the light-dependent reactions of photosynthesis. At the end of the chain, well, it doesn't have to be at the end, but elsewhere in the membrane, we have this very special molecule, which you may have heard of before, called ATP Synthase. Oops, pardon me. There we go. ATP Synthase. ATP Synthase is used to generate ATP from ADP. ATP, remember, is adenosine triphosphate, from ADP, adenosine diphosphate, and inorganic phosphate ions. And the way that ATP synthase does this, because this reaction requires energy, is through the movement of hydrogen ions back down their concentration gradient from where there is a very high concentration in the intermembrane space back into the matrix. Sounds very exciting. This movement causes ATP synthase to actually spin around and generate, that in turn generates ATP. This is where by far the majority of ATP is made in your body, in your cells. Per glucose molecule, we get approximately 32 to 34 molecules of ATP. So this is a very large amount of ATP that is being made by this process. The hydrogen ion concentration gradient then is being maintained by hydrogens being pumped into the intermembrane space. It is actually also being maintained, this concentration difference, by hydrogen ions being used up in the matrix. You see, the electrons have moved all the way down to here. Now they need somewhere to go. And to deal with this problem, we have oxygen. Half a molecule of oxygen reacts with the electrons and a hydrogen ion or two hydrogen ions, sorry, pardon me, to form H2O, water. Water is being made. And for every glucose that is respired, 6 of these are made. 6 H2O. The water can leave the cell, or it can be used by the cell in other processes, but water is being produced here. So, where does the oxygen in water, the water that we produce, come from? It comes from molecular oxygen, the oxygen that we respire. That is used to make water. It is the electron acceptor at the end of the electron transport chain. So a few key points from this. Let's think about what would happen here if we didn't have oxygen. Well, if no oxygen is present, then there is nowhere for these electrons to go, so they're going to stay put. That means that all the other electrons in the electron transport chain are also going to stay put. There'll be no movement of electrons through the electron transport chain, which means that there will be no pumping of hydrogens across the membrane. This means that there will not be a concentration gradient between the hydrogens in the intermembrane space and the matrix, and so ATP synthase will stop to work. ATP will no longer be generated by the electron transport chain. Sorry, folks. This has other consequences too. The two electrons, because they're not being passed on, cannot be replaced from electrons from NADH. So NADH is no longer being converted into NAD+. This means that no NAD+ is available to enable the Kreb cycle to keep turning. You may remember from the Kreb cycle video that there are actually two electron carriers. There is another one called FADH2. One of these is produced per pyruvate molecule in the Kreb cycle. This electron carrier doesn't actually put its electrons right here, instead it donates them further down the chain. So FADH2 carries less energy and is used to pump less hydrogen ions across. But the same problem would happen with FADH2. It would no longer be converted into FAD+, and so it could not return to the Kreb cycle to react. As a result then of a lack of oxygen, the electron transport chain will stop, Kreb cycle will stop, and the only place that you can generate ATP is in glycolysis with the help of fermentation. So, let's look at our totals then. From glycolysis, we get 2 molecules of ATP. And if there is no oxygen present, that is all you get. But if oxygen is present, we also get 2 molecules of ATP from the Kreb cycle and from the electron transport chain between 32 to 34 molecules of ATP. This means that in aerobic respiration we get between 36 and 38 molecules of ATP compared to just 2 from anaerobic respiration. Okay, if you have any questions about this topic, make sure you see me. Great, that's the electron transport chain.